All right, welcome to the second video for Chapter 5. Here we're going to be looking at the three types of passive transport. Passive transport of substances is occurring when no energy or ATP is required for the molecule to move. The molecule is going from high to low concentration down its gradient or with its gradient, and it can either pass through the membrane directly, that phospholipid bilayer, or it might require a transport protein to cross the plasma membrane. We're going to look at three types of passive transport. The first one is known as diffusion, or sometimes simple diffusion. The second one is facilitated diffusion, and the third is osmosis. And all three follow these characteristics above. Several factors will affect how quickly something can cross or diffuse across the membrane. The first is concentration gradients. The greater the difference in concentration on one side compared to the other, the faster the molecules will diffuse. The second is the mass of the molecules. Smaller molecules will diffuse more quickly than larger, heavier molecules. When temperatures are higher, molecules will move faster or diffuse faster across the membrane. Solvent density, or another way to look at this, is dehydration, which will increase the density of the cytoplasm, meaning there are more things in the way, will slow diffusion rates down because the substance will have to go around all of these other barriers to cross the membrane. Solubility is important because the molecules that are more nonpolar will be able to pass that fatty acid layer of the cell membrane more quickly. The greater the surface area of the membrane to cross, the faster the diffusion rate will be. The greater the distance the molecule or substance has to travel, however, the slower the diffusion rate. And finally, in some cells, blood pressure, for example, will affect the diffusion rate. Blood pressure is an example that applies to human kidneys. So kidneys are great at filtering blood, and certain molecules will pass through some of the membranes in the kidneys, and this speed of diffusion or diffusion rate will increase with increased blood pressure. And sometimes we sense this when, for example, if you are nervous or anxious about something and your blood pressure temporarily increases, sometimes you'll feel the urge to go to the restroom, to urinate. Something similar happens when we jump into a swimming pool and we have pressure from that swimming pool water pushing throughout our body. That also increases our blood pressure, and sometimes this can increase the urge to urinate as well. So the first type of passive transport is diffusion, also known as simple diffusion, because we're crossing through or directly through the lipid bilayer. There is no protein channel required. Just like before, I mentioned this means that the molecules or substances are going from high concentration to low concentration down its concentration gradient. And the net movement of the substance will stop or cease once the concentration on either side is equal. There will still be movement, but no net movement of the molecule across the membrane. So what kind of molecules can cross the lipid bilayer without assistance through a protein? These include small, non-polar molecules, and this is because they have to go through that lipid layer of the membrane. And this includes gases like oxygen, carbon dioxide, and small lipid hormones. Looking at those lipid hormones, you might remember from a previous lecture that these include hormones like estrogen and testosterone. So these lipid hormones, because they are hydrophobic and nonpolar, can easily cross the cell membrane. They can easily cross that nonpolar hydrophobic region, and this is the reason, that we can take hormones orally by mouth, or we can put them on the surface of the skin and get them to work through a patch method. These hormones can diffuse and cross through the cell membranes to create their cellular effect. The second type of passive transport is facilitated diffusion, or facilitated transport. This is still passive transport, a type of passive transport, because we're still moving substances or molecules down their concentration gradient, going from high to low concentration. The only difference between facilitated diffusion and simple diffusion is that this time we require proteins to transport the substance, and these are going to be transmembrane integral membrane proteins. The reason for this is because we're going to be looking at the diffusion of small polar water-loving molecules and ions. So because these are charged or polar, they do not mix well with that nonpolar or hydrophobic region of the phospholipid bilayer. That means we need these integral membrane proteins to act as a channel for these molecules to pass. So there are two types of facilitated transport proteins, the channel proteins I just mentioned, or sometimes we use carrier proteins. The most common mistake I see students making is that they confuse this need for proteins as a need for energy as well. So again, there is still no energy or ATP needed because we're going down or with the concentration gradient. Looking at these two types of proteins a little bit in more detail, channel proteins look like a big pore in the membrane, and these are going to be transporting the molecules much more quickly than carrier proteins. Carrier proteins will work more slowly because they actually have to bind to the substance of interest before it can push it across the membrane. So for the channel proteins that move molecules across the membrane more quickly, if I look at their composition, I know that the outer, inner, and pore side, the pore portion or inner core, will be made of water-loving or hydrophilic amino acids because they're going to be facing water. Some of these channel proteins will be open all of the time. So anything that can fit through the hole, the pore size, will be able to cross the membrane, moving the molecule from high to low concentration. Others are gated and they only open in response to some kind of chemical or electrical signal. Some important examples we'll see more later on include aquaporins, and these are specific to water. Water moves through these channels. And later on, you might also see muscle cells have specific gated ion channels that allow for muscle contraction once they are opened through a signaling molecule called acetylcholine. Carrier proteins tend to work more slowly and transport their molecules more slowly across the membrane because they're very specific. Each carrier protein only binds to its specific substance. The binding must occur before the carrier protein changes shape and transports it across the membrane to the other side. And the movement can be in either direction, into or out of the cell. It really depends on that concentration gradient because you're still going from high to low concentration. The differences between carrier proteins and simple diffusion, where something crosses the membrane directly, is that for carrier proteins, you can reach something called saturation, which is when you reach the maximum transport rate because all of the carrier proteins are currently in use. As an example, let's say I have these 3 carrier proteins, and each of them can carry 1 molecule across the membrane per second. So if one of them is only in use, then the current rate is 1 in per second. If 2 of them are currently in use, they're both transporting their molecule at the same time, then the rate would be 2 per second. If all 3 of them are in use, then the rate would be 3 per second. And then let's say I have another molecule in here and I'm trying to get it across the membrane. The max rate would still be 3 in per second because I don't have 4 carrier proteins. All of them are in use and all of them are currently saturated. A common example of a carrier protein inside our cells are glucose transport proteins. Finally, the third type of passive transport is osmosis. And osmosis is the diffusion of water across a membrane where solutes are not able to cross. And you have to be careful here because when we're looking at going down the concentration gradient. We're looking at water going down its own concentration gradient from where there is more water to where there is less water. So this can only happen when we have some kind of selectively permeable membrane where the solutes cannot pass the membrane, but water can. So here I have a picture of my starting container. I'm going to pretend this is some kind of cell, and my red line is that semi-permeable membrane. The purple dots are my solutes, and the solutes cannot cross. The pink solution is my water. So here I can see that there is more water on the left side, high water concentration. On the right side, there is a lower water concentration because there's more solutes. So I'm saying that water is going to go where there's more water to where there's less water. So water is going to cross the membrane, going towards the right side. And that's why at the end of the reaction, I can see the water level has risen on the right side. Another way to phrase this, second bullet point really here, is you can say water moves towards the side with higher solute concentration. I can see that this side has a greater solute concentration and water is moving towards that side. Tonicity is a term we use to describe how an extracellular solution, the solution outside of the cell, can change the volume, really the water volume of a cell by affecting osmosis. So tonicity is correlated to the osmolarity of a solution, where osmolarity is the total solute concentration of a solution, and this includes both permeable and non-permeable solutes. When we're looking at osmosis and water movement, though, it's really the non-permeable solutes that can't cross the membrane that impacts the water movement. So remember, water is always going to be moving towards the side with greater solute concentration. In the next few slides, we're going to look at different types of solute concentrations and environments, including hypertonic, isotonic, and hypotonic, looking at the osmolarity of the environment compared to the cell. A hypotonic environment is when the extracellular fluid, the fluid outside of the cell, has a lower osmolarity or lower solute concentration compared to the inside of the cell. So here they don't show any solutes, but if I were looking at a hypotonic environment, what this means is that there will be more solutes inside this red blood cell than the outside environment. And I know water always goes towards the side with greater solute concentration, so water rushes into the cell, and this causes lysis of the cell. The cell is lysed, so that means the cell bursts. And in an isotonic solution, the environment has the same osmolarity compared to the inside of the cell. So let's pretend I have a certain number or solute concentration outside, and it's equal to the solute concentration inside. So there is no net movement of water into or out of the cell. In contrast, in a hypertonic environment, this means that the outside of the cell would have much greater solute concentration than the inside of the cell. Again, I know water always goes towards the side with greater solute concentration, so this time water leaves the cell. The cell will shrivel up because it's losing water, and this results in something called crenation. The cell crenates. So for our cells or other animal cells, the cells work best when they are in an isotonic solution. Looking at these types of solutions, this also explains why, for example, if we were stuck out in sea, like on a boat in the middle of the ocean, why we could not drink the ocean water. Seawater is more salty, is hypertonic compared to our blood. This means that if we were to drink seawater, water would actually leave our cells even more, making us even more dehydrated and can lead to death. In contrast, this doesn't really happen anymore, but it used to be that when people ran marathons, marathons are, I forget what was it, 26 miles, a little bit more than 26 miles. They used to have water stations almost at every mile. And you would hear these horror stories about people drinking too much water and dying from drinking too much plain water. The reason for this is because our thirst response is slow to start and also slow to stop. So what would happen is if you drink too much water without eating, without consuming salt tablets or something similar, then you create a hypotonic environment and water rushes into your cells, can cause lysis of your red blood cells and death. And in our book, there is a case scenario here where it says a doctor injects a patient with what the doctor thinks is an isotonic saline solution, but the patient dies, and when they look at the red blood cells, they have been destroyed. And our book asks us if we think, actually what we think the solution was that the doctor really injected. We know it wasn't isotonic. What would cause cells to be destroyed? So the cell bursts, is destroyed. That probably was a hypotonic solution. Some living organisms have ways of controlling the effects of osmosis. They can actually counteract water moving into the cell, and we call this osmoregulation. It's really interesting because cell walls in some of these organisms, like plants, fungi, bacteria, and in some protists, like algae, these organisms can resist lysis, the bursting, in hypotonic environments because the wall can push back. That cell wall can push back against the water coming in. So that pressure exerted by the plasma membrane against the cell wall is called turgor pressure, and it's critical to some of these functions of these organisms. And in fact, some organisms like plants actually prefer hypotonic environments. So if I look, this is an isotonic environment where there's no net movement of water into or out of the cell. I can see that the thick black line represents the cell wall, and the inner green line represents the cell membrane. In a hypotonic environment, water rushes into the cell, but the cell does not burst readily because of that cell wall, that rigid wall. In hypertonic environments, you might not be able to notice anything happening to the cell unless you look more carefully. Here, water will leave the cell in a hypertonic environment just as it did for the red blood cell, but in the plant cell or any other kind of cell with a cell wall, the result is not crenation, it's actually known as plasmolysis. The plasma membrane detaches and pulls away from the cell wall, and I can see that here. So if plants are not watered adequately, they'll actually experience plasmolysis, and you'll see a wilting of the plant. You even see a wilting of the plant in isotonic environments. They prefer hypotonic environments where water rushes in. The water causes the plant cell membrane to push against the cell wall, and you see turgor pressure. Even some organisms without cell walls have the ability to osmoregulate. One example is freshwater protists like paramecia, and we saw some of these in the lab the other day, and amoebas. They have large structures called contractile vacuoles that can actually push water out of the cell when water enters, and this allows them to not lyse or not burst. In humans, we osmoregulate because we have osmoreceptors in our brain that monitor our salt concentrations and our solute concentrations in our blood. So one example one way that we experience this is that you may have, after eating salty food, you might feel thirsty. So this causes you to drink more water to lower your solute concentration back to normal. And you might also notice you urinate less when you eat salty food. Fish can do this as well. Fish in fresh water can actually dilute their urine to get rid of extra water or that excess water. Fish that live in salt water can actually produce concentrated urine to get rid of their excess salts. That takes us to the end of part 2. In our third and final video for chapter 5, we'll look at active transport.